Plants have the ability to solve two of the biggest problems we're facing as a humanity. On one hand, they are our food, our nutrients, and the very essence of what sustains life. And in order to feed an increasingly ballooning population that is projected to reach 10 billion people in the coming years, scientists are looking deeper than ever before in order to see how we can produce more food with less resources. But on the other hand, plants are also some of the most crucial engineers. They are constantly grabbing CO2 from the atmosphere and turning it into oxygen, which sustains us, and might hide the solution onto how we can capture more CO2 than ever before. In this episode, I want to take you on a journey with me to show you the incredible past of plant biotechnology and how it changed the course of humanity forever. But at the same time, I want to show you some of the things that we got wrong and how scientists are moving towards a future where we combine just the best of everything we have learned so far. But in order to do that, I need some help.
That's kind of the little compartment within the plant where photosynthesis happens.
This is Dr. Daniel Dunkelmann. He's one of the leading scientists for the Max Planck Institute for Plant Biotechnology, and his lab group is working on developing an artificial chloroplast. This might not mean anything to you right now, but by the end of this video, I promise you will see how incredibly cool this is and why it might be a type of technology that will underline so many other things that we're trying to do with plants right now. But you see, one of the questions that I want to ask Daniel is this technology really any new at all? After all, it's been over 30 years that genetically modified crops have been in our supermarkets.
One of the things that are so essential to understand is that humans, ever since our existence and domestication, have been genetic engineer of plants. So, can't stress this enough. We have engineered plants. We have done it through selection at a time scale that is extremely slow. But we have done that, and what's really important to understand, and I encourage anyone to Google how maize used to look like and go onto the field and see what maize is today.
It might be weird to admit, but indeed, directed evolution and selective breeding are a type of gene editing. It is one that occurs through selection and incredibly long time frames. But since the very first early human societies, we have been trying to make plants better. This is not to make plants some sort of cyborg futuristic plant, but in order to survive. Because even up to a few decades ago, one catastrophic crop yield could mean the difference between life and death for communities. But it might surprise you to hear that the first types of genetically modified organisms were not plants at all, and were not even created in order to feed us. It was this. In 1982, the FDA approves the first genetically modified organism, and it's a type of bacteria that can manufacture real human insulin. This was huge for people with type 1 diabetes that had so far depended on artificial versions, mostly derived from animals, that were simply not as good as real human insulin. But this was incredibly important, as scientists for the first time had a basis to work on and to start tinkering with the instructions of life, DNA.
You have a toolbox that is so well developed. So you can do anything. You can imagine it as a Word kind of text editor. You can copy paste. You now have kind of almost, not yet, but in a few years from now, we'll have a kind of AI-derived tools to help assist write the genetic code, etc. So everything is possible within the prokaryote. You then move to mammalian systems, of which we humans are part of, and there is still a good amount of tools available, and you can really do stuff with precision. And one of the main reasons is also there's a lot of funding that flows into mammalian synthetic biology, because obviously we would like to cure disease and help humanity thrive. But then when you go to plants, you're kind of in a situation, you're almost imagining a text writer and like kind of a text writing machine, finger system. You just have like individual fingers hitting your keyboard. So that's kind of where I feel we are. And
this is Daniel trying to explain to me the difference between different types of genomes. You see, whether we're talking about humans, plants, or our tiny friends, things like fungi, microbes, and bacteria, We all have this universal code of life, DNA. But changing our DNA is not equally easy. And this is where I want to tell you one of the biggest lies we were all taught in school. You see, DNA is not just contained within the cell nucleus, but animals, including humans, have a second type of DNA that is known as mitochondrial DNA. And plants go a step even further and have a third set of DNA. This type of DNA drives the photosynthetic machinery of the plant cell. Okay, I gotta admit, at this moment in the interview, I was feeling pretty okay. I understood that we have 3 different types of DNA in a plant cell, and that differences between different species and different types of cell add an extra level of complexity, with plant biologists getting the shorter stick. But this next thing that Daniel shared with me changed my perception of everything I knew so far.
You can almost imagine that to be an operation at the open heart, because you're really working in the system and you're working in the processes that sustain that system and keep it alive. So in an idealized world, in decades to come, what we could do is develop a completely parallel system to the native system of the organism, where we can play around with without it going directly to the lifeline of the organism.
You see, proteins are the buttons with which we execute every single function in a living organism. For the longest time, we have been tinkering with the DNA that keeps us alive. But what plant scientists are truly trying to do right now is create a parallel system. To go back to Daniel's words, you're not performing open heart surgery, but you're trying to manufacture a second production line alongside the one that keeps you alive. And with that DNA, we can instruct it to make anything we want, whether it is medications, biofuels, plastics, and anything else you can imagine. And to go back to the roots to our GMO quest, some of our most advanced and high-end drugs are also proteins.
So a really good example is like if you have GLP-1 inhibitors, which are currently being used for control of diabetes, but more often than not, I guess weight loss. And so those therapeutics are protein-based, and they're proteins that are modified at 2 specific positions. When you're producing proteins, there is a process that is called solidified phase peptide synthesis. Is a chemical process, and that process has 2 severe disadvantages. One of it is its scalability, so it's quite hard to scale, but people will succeed because there's a lot of money in that. But the much bigger problem is that the chemicals you're using, this process is extremely toxic and wasteful. The dream would be that in a 10-year perspective, we could kind of produce our tomato plant. We could equip this tomato plant with all of the molecular tools to build these modified proteins, and then have a kind of tomato plant that you're growing, you making a powder out of it, and ultimately you're eating it. So you can have your medication from a plant that grows. So more likely than you growing that tomato will be the tomatoes being grown by someone, and this is thoroughly tested, and then you buy your kind of additive that you can add to your food and consume the essential medications.
This one type of technology alone could be absolutely huge for what we're trying to achieve with plant biotechnology. But if you're ready, I want to go one step further, because the most exciting thing that Daniel shared with me is this one tiny, overlooked organelle that we don't even talk or think about, but might just be the most important part in this entire equation.
And this is especially important when it comes to engineering some of the most fundamental molecules of life, amongst which the one that I still think is the most incredible molecule, and this is the protein production machinery, which is the ribosome. And that's a vast molecular machine, and that machine produces your proteins. And so that machine reads all of the mRNAs and reads their code and translates that into all of the proteins. First iteration of any kind of molecular engineering worthwhile doing is probably going to be crap. So what you're doing, equipping with a new function, at the same time you're kind of destroying the old one. and then it can't do anything right. So it can't do the new function properly, it can't do the old function in any kind of sustainable way, which means you're just killing the organism as long as you're working on the lifeline. But if you have orthogonalized the system, you now have a ribosome that's no longer necessary to sustain life. And so what we want to be able in the far future is kind of encode information in DNA that is no longer the DNA that the cell uses.
That is the ribosome. This is also one of the biggest opportunities to fix one of the most important mistakes that we have made in the past with genetically modified organisms, especially crops. The two most common and probably well-deserved criticisms of this type of technology is terminator seeds, or making genetically modified crops that will not be able to yield new seeds every single season, and farmers depend on these corporations to continuously supply them with seeds. But the second, and perhaps most important, is the fact that these artificial genes that we're planting out there in nature might find their way back into our natural wild-type plants, and thus destroy some of the natural diversity that we're so hard trying to protect. And this is where Daniel went into incredible detail, explaining to me how this new era of plant genetic engineering is going to start with safety mechanisms first.
The long-term vision that also my lab and other labs are pursuing is that we want to write codes in these orthogonal genetic codes that are genetically isolated, which means we can then play around with them at will and have no longer the fear that we're cross-contaminating the natural world. And then we can go a step further and build in kill switches, and we can really add layers and layers of control through engineering.
In simple words, we are creating artificial plant parts that can do all the things we want them to, whether it is create new drugs or capture more CO2. Scientists are now working towards an era of creating plants in a way that we have never seen before. But this time, they're able to design the systems and these plants with more safety mechanisms and more effective control.